Welcome to Brain Ponderings, your window into the great world of neuroscience research, and conversations with experts in their particular area. And today it's my great pleasure to welcome Professor David Gutmann. He's the Donald Schnuck Family Professor of Neurology and Director of the Washington University Neurofibromatosis Center. We'll talk a lot about neurofibromatosis. He's one of the world's experts. But more broadly, David is an expert and is doing a lot of fascinating work on how cells in the environment of cancer cells affect the cancer cells and vice versa, how the cancer cells sometimes, say, co-opt or take advantage of some of the signaling mechanisms of the cancer cells. So welcome, David.
Well, thank you for inviting me. This would be fun, and we'll look forward to chatting you more about things that are going on in the world of cancer neuroscience.
Yeah, I'm really interested in this. I had a actually a PhD student. I don't know if you've come across him, Justin Lathia.
Oh yeah,
Yeah, he's up at Cleveland Clinic, but in my lab, he kind of did this joint thing with Cambridge, developmental neurobiologist in Cambridge, and then he did most of his research in my lab at NIH on neural stem cells, and particularly the neural stem cell niche and how signals around that. And then now he's kind of full force brain cancer, glioblastoma, which is one type of brain cancer. You have a very interesting background. You trained in some people's lab who I know and were my ultimate boss at one time, Francis Collins. And so why don't you talk about your background? You grew up in Michigan, right?
absolutely. So I was a Detroit boy. I grew up in the shadow of the University of Michigan, so it was a natural lure for me to go there as a newly indoctrinated Wolverine. So I spent my undergraduate, graduate school, medical school, and eventually went back for a postdoctoral fellowship all at the University of Michigan. One of the nice parts about Michigan for me was just the flat academic landscape, that there you had opportunities to work and be exposed to a large number of people with diverse vantage points and skill sets, and it really helped me become the kind of scientist that I wanted to become. I, as a and early on in my training at the University of Michigan as a medical student, I found myself quite disappointed with the didactic, dry discussions that went on, and uh, the amount of purge of binging and purging that goes on in terms of how academic knowledge is provided and then retested. And at the end of my first year of medical school, I went to the academic advisor and said, "I am dying here intellectually. Can I, go off and work in a lab?" And they said, "Well, whoa, whoa, whoa! You have to take classes, and you have to, still pass the exams." And I said, "Well, how about if I do both at the same time?" And they were very gracious and let me do a master's in the Department of Genetics. And one of the pioneers in human genetics was our chair, which was Jim Neal. Jim Neal was, at many people's eyes, the godfather of human genetics. And he really turned me on to the power of genetics and to understand human disease. And he pointed me to a professor who was looking at genetics to understand immune cell interactions. And that was John Niederhuber, who eventually was lured to the National Cancer Institute to be NCI chief. And John inspired me as a physician scientist, gave me my first start working on the genetics of T cell macrophage interactions, and finished my PhD with him, finished medical school, and fell in love with neurology and went off to the University of Pennsylvania as a neurologist in training. And there I fell under the spell of Kurt Fischbeck, another NINDS and NIH eventual transplant. And Kurt said, well, if you're doing all this genetics of immunology, have you considered genetics of neurologic disease? And so we worked together when he was in the early stages of working on Kennedy syndrome. And, I was just sort of lucky enough to learn things like how to do PCR reactions in his lab. And at the end of my training, he turned to me and he said, I was at this grant review meeting in New York City, and I met this guy. He's not done a lot yet, but he seems incredibly brilliant. He's working on the genetics of neurologic disease. And you really should give him a call because he'd be a great place for you to do a fellowship. So I initially emailed and then called this guy named Francis Collins. And, Francis said, oh, that would be great. We're looking, I haven't had a neurologist in the lab before. Why don't you come up and interview? And I interviewed, and, he took me on as uh, a fellow, research fellow in his lab after I finished residency. And what
That,
Francis said
that,
to me, yeah,
that back at Michigan.
that's back in Michigan.
Yeah.
So spent a long time at that University of Michigan. And then obviously Francis in the last few years of my training with him went off to become initially genome czar and then NIH director many years later. And what I learned from Francis was how truly to be a physician scientist, how to be outstanding as a physician, somebody I could come to clinic with and just watch him interact with patients and the admiration and the respect that his patients had for him was a bar that I wanted to reach. And then go to the laboratory and have ideas that just mere mortals didn't have. And to sit there at lab meeting as we describe for him for the first time something we were struggling with and have him sit back in his chair and go, well, have you considered? And then blam, it was so obvious. And I said, I'm going to be like Francis Collins when I grow up. And, Francis was generous enough when I came to the lab as a neurologist. He said, I have these diseases that we study. We have Huntington's disease and we have neurofibromatosis. And I'd suggest you work on neurofibromatosis. I think we have the gene. And and if you come to the lab, everybody else in the lab wants to work on the DNA and the RNA, but you might be interested in the protein and its function. And I said, where do I sign, Dr. Collins? And then that I fell in love with the NF, both patients and the research, and the rest they say is history.
And when did you first see someone with neurofibromatosis, and when, and then, so what was kind of your main findings in your time with Francis?
So they answer to your first question is that I first saw patients with usually kids with neurofibromatosis because Kurt Fischbeck recognized I was interested in genetics, so he sent me over to Children's Hospital of Philadelphia, and they had a very large NF clinic. So I spent a lot of time seeing those kiddos. So when I came to Francis's lab, I felt like I had already drunk the Kool-Aid. I was ready for the adventure. And what I did
Ah,
in Francis's lab for the first year was try to identify the protein. We've spent a lot of time generating antibodies. Finally was able to identify the protein, which we published about 9 months or a year after I joined the laboratory. And then he said, well, after you figure out what the protein looks like, we got to figure out what it does. And there he just let me loose. And it's one of the beauty of studying in a lab like Francis Collins is he said, whoever you need to call to learn more about how it functions, just call them. And, he put me in touch with some of the gurus in RAS biology, Mike Wigler and Daphne Barsagi, and Julian Downward, and, allowed me to play with them in their laboratories to learn more about how the NF1 protein functions.
and can you describe, there's so many different manifestations of this particular disorder. Can you just kind of describe neurofibromatosis?
Absolutely. So it's a genetic disorder. So about half of the folks that I see in clinic have a family member who has NF and the other half don't. And when you examine these kids, you find that they have birthmarks, and that's the birthmarks are often termed cafe au lait spots, and that's the most common feature. That's how they come to medical recognition. But kids with NF1 are at significant risk for developing learning, cognitive, and behavioral problems, including autism, and they're at elevated risk for developing tumors, including tumors of the nerve called neurofibromas, and tumors of the brain like brainstem gliomas and optic gliomas. And
And okay, and what is this missense mutations or what are, one amino acid different in the protein or?
it's a great question. So it turns out that one of the puzzling aspects of NF is that the mutations vary from patient to patient, and there are relatively few hotspots, which makes it difficult to do genotype phenotype correlations. And so, of the tens of thousands of folks that have had genetic testing, there are thousands of different mutations. And one of the ideas that we have in the lab is that the type of mutation, where that mutation is, what that mutation does to the protein, does have an effect on whether or not you'll have learning problems, brain tumors, and maybe one of the predictive markers for the complexity of the disease.
All right. So, well, sometimes, for example, yeah, there are a lot of mutations. There are hot spots, particularly, well, and usually in those, and I guess I don't know this, so I'll ask you.
Uh-huh.
Um, my experience is that you're more likely to have gain of function mutation rather than if you have mutations that are kind of scattered all over, maybe loss of function. And these are loss of function mutations.
That's right. They're all loss of function. And the only thing that we knew for the first 20 years of NF research was that one portion of the NF1 protein acts as a suppressor of RAS activity. What the other 90% does is still a little bit of a
Oh,
mystery.
I see.
But all of the mutations that we or others have examined lead to a protein that can't regulate RAS anymore. So it's not as simple as RAS. And I think what having different mutations confers is effects on different signaling pathways or different protein interactions. And that may be another determinant of the variability that we see.
So is NF1 it's in the cytoplasm of the cells, or where's it
For the
located?
most part, it lives in the cytoplasm, and when it's needed to shut off RAS activity, it gets recruited to the membrane, where it can interact with the signaling molecules that initiate RAS activation. So it
And
does a
so,
little bit of shuttling.
uh, yeah, my son just got his PhD in January in biochemistry from Hopkins, and he was doing this. They have this neat way using biotin coupled antibodies to try to pull out all of the proteins from whatever cells you want that interact with a specific protein. My son was studying a protein called dynactin.
Mhm.
And so what other proteins, I'm sure by now that's kind of thing's been done with NF1.
Yeah, so one of the challenges for NF1 biologists is that the protein is rather large. It's 2,818 amino acids,
Oh,
so it's
wow.
a fairly big protein. Up until recently, maybe the last 5 or 6 years, it's been difficult to express the whole protein without it falling apart or not being expressed or acquiring mutations. So some groups, Frank McCormick in particular, went through the arduous task of changing the nucleotides to preserve the amino acid, uh, but to change it so that it was no longer degraded and could be reassembled as a full-length protein. So now we're starting to do those kinds of studies. What we know so far is that the NF1 protein called neurofibromin interacts with a protein called spread1, which is interesting because that's mutated in an NF1 related disorder called Leach's syndrome. And so that turns out that interaction brings it to the cell membrane. It also interacts with tubulin, and it may interact with other proteins that are important for neuronal function, and that's where we don't know whether or not those protein interactions are key to its activity.
So it's um, what about interactions between, so do all cells express neurofibromin
At
and
different levels.
different levels? And is it in only in proliferative cells that the mutations have a adverse effect?
So it turns out that NF1 mutation leads to neuronal dysfunction.
Yeah.
It leads to oligodendrocyte dysfunction when there's simply one mutation. And if both copies are lost, then in some cell types that leads to increased growth and tumor formation.
Huh.
So it's very cell dependent and context dependent, which is, one of the challenges and one of the beauties of studying NF1 is we still have a lot of the protein still to solve in terms of what it does. And there's a tremendous amount of diversity depending on the cell type or even the developmental stage that you're examining.
So you mentioned, early on, kind of your background in immunology, genetics, and then cancer. And so how do you, what kind of models are you using? Do you have animal models that you can, manipulate different things and try to get down to what's actually going on in the living animal?
Absolutely. So there are multiple types of models that we built over the last several decades. One is what are genetically engineered mice that develop brain tumors. So we developed the first NF1 optic glioma model where the mice develop these brain tumors along their optic nerve. We also use actual patient operative specimens. So we'll generate tumor cell lines from the patients that we can re-engraft into recipient mice and study. And then we also use human-induced pluripotent stem cell engineering to uh, look at the effect of NF1 mutation on human neurons, as well as in mini brains, organoids, and assembloids, as another avatar um, for how to study and how to screen for drugs that might ameliorate the condition.
So the optic nerve gliomas, two questions. Do they become malignant? And the second is, do they affect vision?
Absolutely. So the answer to the first question is fascinating. We rarely, if ever, see these become aggressive. So to me, they feel much more like developmental abnormalities, even though they're in the cancer realm, because they have a peak in early childhood, and then it's incredibly rare to see these tumors progress in teenage kids, and never see optic gliomas, or almost never see optic gliomas in adults, and they certainly don't come back as malignant counterparts. And then because of its proximity to the nerve, the tumors do cause vision loss, which is one of the significant problems that our kiddos face. And these are usually young kids, as I mentioned. And so these kids may start to lose vision between 3 and 5 years of age, which severely limits their ability to perform in school. And as teenagers, limits their employment opportunities and their abilities to drive.
Now, there's been some, maybe we'll have time to talk about it, but big advances in gene therapy approaches, right? And particularly in the eye, because you can like inject things right
Right.
right into the eye is the if you inject say I don't know if you could do this because the gene's so big for this neurofibromatosis gene. You know, CRISPR, could you correct the gene defect and stop the growth of the cells or some kind of, is there any translational work going on?
Absolutely. So there's three ways that people are thinking about it. Some are most of these are operating in other labs, but I'll tell you about the ones that are in other labs as well as our own lab. One is to use cell transplantation. So can you, because the neurons are dying and
Oh,
you want them to regrow and form connections, can you put in cells that have the capability of forming new retinal ganglion cells as a therapy? The second would be genetic engineering, which is great if you know what the mutation is and if you
yeah.
can CRISPR edit. And then the third, which is really where we focus, is to try to understand why the neurons die and interrupt that death signal. And, one of the things that we showed a number of years ago is that girls tend to lose vision more often than boys who have optic nerve gliomas. And in mice, that's absolutely true. It's almost a completely sexually dimorphic phenotype in mice. Both male and female mice develop optic gliomas, but the female mice are the ones that lose vision. And that's allowed a former postdoctoral fellow in my lab, Caroline Tang, who's a neuro-ophthalmologist. She's been repurposing drugs like Lupron, which we
Oh.
use to suppress the gonadal axis in kids who are developing early onset puberty, to suppress the death signals that are coming uh, from the area around the tumor and causing the neurons to die. So we think the other strategy would be pharmacologic rather than cell therapy or gene therapy based.
And is there a lot of inflammation? There must be inflammation going on in association with degeneration.
Yeah. So maybe I'll take a couple steps back and we'll
Okay.
we'll we'll talk about that inflammation in a slightly different context. So early on, based on the idea that tumors cells lose both copies of the NF1 gene, we developed a mouse that both copies of the NF1 gene are inactivated in the cells that should give rise to the glial compartment in the optic nerve. And we were surprised when those mice never developed tumors. And so we then said, well, that's not the same genetic makeup that patients have. Patients have a germline mutation. So there's an NF1 mutation in every cell in the body, but the cells that become tumors lose both copies. And so we generated that mouse, and that mouse almost 100% of the time develops optic gliomas. And what that said to us is those other cells are really important for generating the tumors. And that's what got us to look at immune system cells, like you said, and then further down the line, neurons. And so while we don't think there's inflammation in the classic sense, we have shown over the last decade or so that T cells are required for the growth of the tumors because they talk to microglia in the brain, and the microglia produce compounds that allow the tumor cells to survive. So that's gotten us to rethink tumors as circuits, that there are cellular circuits. Some of those cells are non-neoplastic cells or normal cells that have an NF1 mutation. And then there are these cells that are the cancer cells. But those circuits are really what maintain the tumor and can be interrupted to block tumor formation and growth.
Okay, so what more specifically, what are the circuits? So in the optic nerve, I guess we'll stick with that for now.
Sure.
So you have the axons from the retinal ganglion cells that are going back to the back of the brain. You have oligodendrocyte progenitor cells that form the myelinating cells.
That's
There are astrocytes there. There are microglia.
Mhm.
Um, Anything else?
And
And the
infrequent T
and
cells.
and the huh.
So we think
Okay,
there's
and
at
then
least three
so
circuits.
So what are some of the signals, right?
Yep.
I know quite a bit about signals between neurons and glia. And actually the microglia are they're an innate immune cell,
Right.
similar to peripheral macrophages. So they respond to infection or injury. And one of the things is to clean things up, for
Mhm.
example, in wound healing. Um, but
So we think there are at least three circuits. Two circuits we've characterized pretty well, and one we're in the process of characterizing. So I'll tell you about the two that we know the best. And one of them is a neuron T cell microglia cancer circuit. And so that circuit is created by a mutation in the NF1 gene that operates at the level of the retinal ganglion cell, causing that retinal ganglion cell to be a little more active, fires more frequently, and its firing leads to the release of a signaling molecule called midkine that acts on T cells to make a chemokine that activates the microglia to produce a growth factor for the tumor cells, and either disrupting the neurons, disrupting the T cells, or disrupting the microglia all lead to cessation of tumor growth in mice.
and as I have a background in developmental neurobiology, and there's a lot of evidence supporting this idea of activity-dependent release of neurotrophic factors.
Mhm.
So if you get hyperexcitability of those retinal ganglion cells, they're probably releasing a number of different factors, just in the absence of a NF1 mutation. And they're probably serving some normal function in maybe regulating myelination or the growth of the axon or even survival of the cells. And so do you think that it's just the cancer cells have the systems to respond to these same factors that would otherwise be doing a good thing in service of the neurons?
Absolutely. So let me break that into sort of two answers. One answer is yes, there is an activity dependence. In the first circuit that I described, we don't know whether it's transcription or translation. That's what we're trying to figure out now, but it's clearly activity dependent. The second circuit is exactly the one that you described, was collaborative work with Michel Manger at Stanford. And what we were able to show in a couple of papers was that the activity in the neurons changes the dynamics between the oligodendrocyte precursors and the neurons, and that when the neuron is active and it becomes active through by light stimulation, it now releases an enzyme that cleaves off a growth factor that's on the surface of the oligodendrocyte precursor, allowing the tumors to initiate and form. And that activity dependent uh, stimulation of that particular circuit uh, can be abrogated by raising the mice in the dark. And so, in a really cool set of experiments that we did together with Michelle, if you dark rear mice that would develop an optic glioma in the dark for 6 weeks, starting soon after weaning, they never develop tumors. And that's because we broke that activity dependent circuit between neurons and oligodendrocyte precursors.
Since since a NF1 is a negative regulator of RAS, which is a pathway activated by a number of different growth factors, then um, so its normal function may be to limit the growth of the normal cells
Mhm.
in some way, keep things with. Now, these are these glioma they're transformed, right?
Mhm.
And so what other, usually you would have to have some other mutation in combination, or at least the little I know about cancer. Uh, for example, most cancers have, or many have mutation in P53 or other tumor suppressor. genes.
The setting of NF1, it's true in the patient samples as well. If you look at the patient tumors, they're pretty bland genetically. And so if you, the vast majority of the tumors have only biallelic NF1 loss. So they don't have P53 mutations, or they don't have PTEN or loss or EGF receptor amplification that you would see in a high-grade tumor. However, should a patient develop a malignant glioma in the context of NF1, they will have some of those other genetic changes. So the low-grade tumors seem to be one, a lot more dependent on their microenvironment for signals and for support, uh, but are genetically pretty simple in terms of their etiologies.
Now, glioblastoma is a brain tumor we hear a lot about
Mhm.
because it's very devastating and has almost always a rapid course that once a person is diagnosed, just in a few years, they die and there's a lot of work going on it, but no truly effective treatments. And one area that's, I guess, has been really important in the cancer field for quite a while now, but it's the idea that within a tumor, all the cancer cells are not equal, and that there's a subpopulation of a tumor where what are called cancer stem cells that, for whatever reason, seem to be more resistant to being killed by a treatment, whether it's chemotherapy or it's some drug. And so, well, with glioblastoma, a lot of times they do surgery, but um, and these stem cells seem to be what's surviving and often giving rise to reoccurrence of the cancer cell. So what's going on in the tumor environment? You have different cancer cells with different phenotypes, and I guess cell survival, proliferation potential, and then you have all, it's very complicated, right? You have all these cells around it, different glial cells, neurons, and so on.
So in in the malignant tumors, there's a lot of cellular heterogeneity. So, and they may not all come from the same cell. There may be multiple different clonal origins for a glioblastoma, even within the same tumor. And like you said, there's enough cellular plasticity, whether it's moving from a state that looks more like an astrocyte to one that looks more like an oligodendrocyte or reverts back to a stem cell, than we see in the low-grade tumors. The low-grade tumors are slow growing for one. Um, at least from our work and some of the work of others, they may come from a single cell. They're monoclonal as opposed to polyclonal. Uh, and the only thing that really is different is the tissue architecture. If there's a stem cell niche, it's not around a hypoxic area like it is in the glioblastomas.
Hmm.
So some of the rules may be different, and it may be because the low-grade tumors are just aberrations of normal development. It's one way to think about it, as opposed to the way we conceptualize cancer, which is a series of unfortunate events that leads to something that's unrecognizable as a normal cell.
Now, if you take some type of any type of cancer cell, I guess, and if you overexpress increased levels of NF1, um, will that have any impact on them?
So we haven't overexpressed all the entire protein, but if
Ah,
you either correct the RAS dysfunction, and there are drugs that we are now in the clinical workspace called MEK inhibitors that are downstream of RAS, you can suppress the growth of the cells. What we don't know either in experimental model systems or in people is whether that's a permanent effect. So, what happens when you take a patient off of a MEK inhibitor? Will that tumor continue to grow, or will it simply say, I'm done with my growth spurt and I'm tired. And in some situations in other tumors, when you take those MEK inhibitors away, the tumor actually rebounds and grows a little bit faster. So that we don't know yet how long to treat and whether we're just suppressing cell growth
Yeah.
or permanently disabling it. And that's why I think alternative strategies that look at the tumor microenvironment may be very helpful because they really target the support that the tumor cells need, as opposed to trying to put our foot down on the tumor cell, because it may just decide it wants to grow when I remove my foot.
Now, you mentioned the increased activity in the retinal ganglion cells will promote the growth of the glioma, and all neurons in the brain have receptors for glutamate, which is excitatory. It'll depolarize,
Mhm.
get calcium influx. And can you talk a little bit about this paper, which actually I just saw it this morning when I was looking over it, looking through Neuron, the journal Neuron. You had this interesting paper. Now, glutamate receptors, the ones that have been studied the most are ion channels, either sodium channels or calcium channels. And there's also another type of glutamate receptor that's well known, coupled to G proteins and signaling. But you apparently found this really interesting role for glutamate in controlling these low-grade gliomas.
Absolutely. So what we were interested in, building on the work of Michel Mange and Frank Winkler, is how does glutamate control tumor cell growth? So they have focused on malignant gliomas, where they've shown that they form synapses and that there's an electrical coupling between the tumor cells and the neuron. What we were interested in is it physical coupling, electrical coupling, or maybe a repurposing of normal physiologic relationships between the cells that give rise to the tumors, which are oligodendrocyte precursors, and the neurons. So neuronal activity leads to the elaboration of glutamate, which instructs the oligodendrocyte precursors to proliferate or differentiate, but it also releases platelet-derived growth factor, and that functions as a stimulus for the proliferation of the oligodendrocyte precursors. And so we look to see whether or not the glutamate that's released by the neurons acted on the tumor cells to electrically couple them so that the calcium influx would lead to increased proliferation. And we were surprised to find that there was no increase in electrical membrane activity, but rather there was an increase in cell growth and in downstream RAS signaling, which would be MAP kinase or MEK signaling. And what Karina Anastastaki, who's a research assistant professor here at WashU, showed is that when glutamate hits the pilocytic astrocytoma, these low tumors, it couples the glutamate receptor to the platelet-derived growth factor receptor. And instead of providing both a differentiation and a proliferative signal, it says we're going to subvert those two receptors to drive cell proliferation.
Huh.
So glutamate through the glutamate receptor activates the platelet-derived growth factor receptor and stimulates cell growth. So we can now have several new pharmacologic targets. One obviously would be to repurpose drugs like memantine that we use to block glutamate receptors, but also blocking the platelet-derived growth factor receptor um, is just as effective um, as as blocking the glutamate receptor. And we think this is just the tumor cell being clever at usurping normal interactions with the neurons for its own selfish purposes to grow.
Yeah. And since The thing with glutamate and drugs, though, is it's so important for everything the brain does
Mhm.
that manipulating it a little bit, maybe you can have some benefit, but if you inhibit too much, then you got big problems. You can't learn and remember things
That's right.
and so on. And with memantine, which it is prescribed to patients with Alzheimer's disease quite a bit, and it had a small but significant. Uh, Actually, it's the only drug so far that's had a beneficial effect in slowing the course of the disease. It's not dramatic, but it's there.
Yeah.
But if you get the dose too high, it'll cause like psychotic episodes, kind of like ketamine, which ketamine, as far as we know, does pretty much the same thing as memantine does on the NMDA glutamate receptor. Um, yeah, so
So I agree with you. Not, it's a right now where we're focused is this a common theme for
yeah, yeah,
cancers?
yeah,
Um, Ultimately, where we'd love to go is in the clinical workplace. Can we use that? And our colleagues in Germany are using drugs that target the glutamate receptor for patients with high-grade gliomas because they're you're talking about a much more dire situation, and we really do need to try to find alternative therapies. Where we're focusing our efforts is that first circuit, which is that I described that has the neuron activity that's caused by NF1 mutation, activating that immune axis. And what Karina Anastasaki showed in those studies is that that electrical activity that's induced by NF1 mutation is inhibitable by lamotrigine. And what she went on to show is that you can use lamotrigine to silence the activity of those NF mutant neurons, that they no longer make the factor that's necessary for T cell activation. And then in mice, you can actually treat mice before the tumors form for a period of time and then stop treating them, and they'll never form a tumor even months later. And so, where this has led us is a clinical trial that we hope will launch sometime in early 2026 to use lamotrigine as a first in its kind chemo preventative agent for optic gliomas in children with NF1. And that's where we're really focusing our energies is, can we push the envelope by using drugs that are safe to use in children,
yeah.
um, to interrupt that process of tumor evolution, and maybe either by itself or in combination with other drugs, really move the needle for how we manage these kids.
Yeah, I was thinking of this, so reducing excitability, obviously little kids often have epileptic seizures,
Hmm.
but
Absolutely.
just in general, and then usually they disappear and they never have them again. But and there's a lot of drugs that are used to treat epilepsy, and they act as quieting
Hmm.
the neurons. And so, are there any clinical data? First of all, what's the prevalence? How common are these mutations in NF1? And are they common enough that one can ask, do some of these kids have seizures and were treated, and then, you know, later on they have less adverse effect, or is there so much variability and other confounds that it's hard to know?
We did look, and it turns out that not many kids at any one institution are treated with lamotrigine. The reason we did not look at the other anti-epileptics was in Karina's early studies, at least in mouse retinal ganglion cells with an NF1 mutation, the effects of other anti-epileptics or mood stabilizers was not equivalent to lamotrigine. So
Okay.
lamotrigine and rufinamide, which are sort of kissing cousins, had a very similar effect at suppressing activity. Some of the other ones actually increased activity, and so they were taken off of our discussion plate early on.
I see. I see. Okay. And let's see, what else? Um, you mentioned that kids with NF1 mutations often have neurological disability, whether it's cognition or my understanding is they often have reduced IQ and maybe some behaviors that are similar to autism or ADHD. Is there any work going on? I mean, do you think that's just from the consequences of the mutations in neurons per se, or could be other cell types?
Yeah, so it's a really interesting idea. We've been very neuronocentric in the NF field, ascribing all of the cognitive problems and the behavioral problems purely to neurons. And in a study that we did with Michel Mange, what we were able to show is that some motor learning tasks are actually driven by NF1 mutation in oligodendrocytes, not in the neurons. And so that relationship between neurons and oligodendrocytes is key for normal uh, normal learning and behavior. There's a lot of work that's just beginning. There's a private foundation called Penny's Flight that has been sponsoring a meeting that I organized initially last year and um, was repeated again this year at the Banbury Center at Cold Spring Harbor. And that group is focused on trying to understand how to categorize learning disabilities in folks with NF1, how to use the models that exist to to find new drugs, and how what's the framework, what's the clinical trials that we need to have in order to test those drugs. And so that group is called Cognition and Behavior in NF. So the cabin group is now working on a second sort of position paper describing where we need to go and to sort of change how we're approaching attention deficit, learning problems, and social interaction, autism-like phenotypes.
All right, so you're the director of the Neurofibromatosis Center
Mhm.
there at WashU, and I think from all you've said, we get kind of a vague picture of exactly what the center is comprised of in terms of clinicians, basic scientists. Do you want to spend a few minutes talking about the center?
I would love to, because I am so proud of the people that I have the opportunity to work with here. We have an amazing group. So there are three pillars to the NF Center. One is the clinical enterprise, the research enterprise, and then what we call really habilitation. And I'll explain each one of those. So in the clinical group, we're one of the very few centers that has 4 clinicians on the pediatric side and 4 clinicians on the adult side, and an active adolescent young adult transition program. And so we really feel that we need to be able to span all of the problems in NF from the first time we see a child way into adulthood. It's also an opportunity for us to see how NF evolves from childhood into adulthood, because historically they're kept separately. And so we don't really understand what happens to an optic glioma or brainstem glioma in an adult because they're not followed by the same team. The research group has 10 laboratories that are focused on different aspects of NF. And embedded in that is a partnership with Philip Payne, who's our Institute for Informatics director, using AI and natural language processing to develop predictive algorithms for uh, for NF clinical features. And then the third part is part habilitation and part education. And in the education, we run a clinical program for folks from other countries to come in and learn about neuro-oncology and NF. That's with my partner, Mohammed Al-Baldaki in pediatric heme onc. We also um, had all of the pediatric residents spend 2 weeks with us so that when they go out into the community, everybody knows what NF is because they had to spend a couple of weeks with us in NF clinic. And then we also have a virtual NF tumor board where there are 35 different countries with 35 institutes within 25 different countries that will present their difficult patient situations to us for us to discuss. And then the other part, that the habilitation part, is that we realize that we focus a lot in the clinic on what kids can't do. We talk a lot about what their disabilities are,
Yeah, yeah,
tumors, vision, seizures, whatever it is, but we don't spend a lot of time emphasizing what they can do. And so about 15 years ago, we developed uh, a number of out-of-hospital programs that are therapy-based, but are really fun for the kids. So we partner with local institutions like the zoo and the science center and the botanical garden, so that we provide therapy in a fun situation for our kids. Uh, they're getting their therapy one-on-one, the parents are networking, and we're really focusing on what they can do, less on what they can't.
yeah. That's fantastic. That yeah, the parents networking. Of course, I'm sure you have parents and kids from uh, quite a distance, but now so they can get together and talk about the issues and share their stories and just kind of appreciate what these kids can do.
Yeah.
And yeah, and right. So do they do they respond, say the ones with the disabilities, intellectual disabilities, do they respond to any kind of cognitive, do they do they learn certain things better than other things, and may they not have, isn't it possible they have some particular skills or ability to learn some things but not others, so that what you're really seeing is, they can excel in some areas, maybe other areas not so
Yeah.
much.
So as a general rule, if you sort of looked across, say, 100 kids, about 25 would be on the attention desk, would be on the spectrum for autism. About two-thirds would have attention deficit, and maybe half have some form of learning disability. But what we don't know, and hardest, one of the challenges for us as a field, is defining what the phenotypes are, and are there endophenotypes, subgroups of kids that we have not identified that might respond uniquely to behavioral therapies, medications, or other um, uh, adjuvant therapies in the classroom setting, and that's really where this cabin group is focusing its attention on, is what is it? What are the disabilities?
Yeah.
And once we can define them, uniquely in a large population of kids with NF, then I think we're in an opportunity to model, because we know what we're modeling, and then we'll be able to use that information to develop maybe subgroup specific treatments that will actually work for the kids instead of treating their symptoms, which is where we are now.
I can't remember if you said this or not, or if I just got it from reading one of your articles, but is it like 1 in 1,000 kids? Is that
It's about 1 in 2,800 kids, so
one
it's
in
a little less
20?
than 1 in 3,000. So
So
almost everybody has met somebody with NF.
yeah,
So it's pretty common, but still in the rare disease category.
yeah, yeah. And what what is NIH doing? I mean, most of your work supported by NIH, I
Mhm.
assume.
Yeah.
What about private foundations? Certainly with it, it's rare, but not so uncommon. Um, what private foundations are there that support research in that area?
So you're absolutely right that the major funding has come from the NIH or the National Cancer Institute. A good amount of funding, almost about 25 million a year, came from the congressionally mandated medical research program through the Department of Defense. That's on pause for this year, but hopefully we'll start again. That particular funding was instrumental in starting a NF clinical trials consortium 20 years ago, which is the only clinical trials consortium focused only on NF. So all of the clinical trials go through that DoD supported mechanism.
And
The,
is that so DOD? So the reason for that was because the military personnel's kids are what I mean are was
the way I understand it, Congress can direct some funds to medical research through the Department of Defense.
I see.
And there was a representative from Utah many years ago who had
Uh,
a child with NF.
yeah.
And that was that, among other things, including tuberous sclerosis and autism, became a funding priority for these Department of Defense initiatives. And they have really been incredibly generous. And we would not be in the position with clinical trials without the Department of Defense.
Yeah,
And
interesting.
then the next rung are really uh, either foundation support. So a major funder of NF research right now is the Gilbert Family Foundation. They've been incredibly visionary and supportive of high risk science, both in the realm of neurofibromas, optic gliomas, malignant brain tumors, and also in behavior and cognitive problems. And they've been a major leader. And then there are other smaller foundations that are just beginning to crop up over the past several years that really speaks to the power of the parents. And one of those is Penny's Flight Foundation that was started a number of years ago. A young lady with NF1 passed away early in her life, and her parents decided that the way to honor her was to start a foundation that was really going to help change the way we practice. And they're the ones that sponsored the cabin meeting at the Cold
Oh,
Spring Harbor. And so I think we're seeing a little bit more of that these days, with the changes that are happening in federal funding currently.
and of course, any new discoveries that are made on NF1 are there's a good chance they're going to be applicable to other cancers, or you may discover new basic mechanisms of how the brain works, interactions between cells. So there's so much potential for, I mean, the focus is on one particular genetic disorder, but on the other hand, it's very likely because cells are very complicated, but there's so much, there's a lot of commonality when you start to get down to how they actually signal between each other.
Absolutely. And I think NF1 is a single gene disorder that causes a lot of common medical problems like autism, uh, becomes a poster child for how to study a disorder like autism or brain tumors by using the lessons from NF1. And we've already seen how that's transforming the landscape for other disciplines besides NF1.
Yeah, and then you can draw on other people too. That's the way
Exactly.
science works. That's why it's so great.
That's
Uh,
right. Yeah.
and okay, so I appreciate David you taking the time for this. I know you're much more busy than I am now. You got a big group there and a lot of responsibilities. So I'll let you go.
It was my pleasure, Mark. Thanks so much for inviting me.
Yeah. Bye.
Bye bye.